Hello everyone, my name is Panagiotis Chrysinas and I'm a PhD student at the University of Buffalo at the Department of Chemical and Biological Engineering. Today, I'm going to present on cross-tissue drug signature predictions for drug repurposing. Drug development is a lengthy process with a very low rate of success. In general, it takes more than 10 years and over $1 billion just to produce one FDA-approved drug. That gives little incentive to the pharmaceutical industry to invest on drugs for rare or orphan diseases, which are currently more than 7,000. A notable workaround to that issue is the so-called drug repurposing, that is, finding new therapeutic use of drugs that differs from the original indications. Data-driven approaches can help in narrowing down the list of potential therapeutic compounds for repurposing, enabled by the generation of large datasets of drug response, such as the connectivity map that contains more than 1.5 billion molecular signatures for 71 different cell lines and 20,000 compounds. Although impressive in size, these signatures are taken mostly from immortalized cancer cell lines, whereas data-intensive approaches for drug repurposing require data from the specific human tissue. We drew motivation from the above challenge and we developed imputation strategies for predicting transition signatures of drug response in target cell lines using data from other, or as we call them, reference cell lines. In the part A of the figure, we can see the main goal of the study, that is to infer drug cell response in target primary cells, are denoted with a red color, by combining data of reference cancer cell lines, denoted with a blue color, and the namely background data, which are denoted with a green color. Now, for this study, we define the background data as the data from the target cell line that do not have any overlapping conditions with data from the reference dataset. In order to achieve the inference, we have developed a two-step imputation strategy, as we can see in the part B of the figure. In step 1, we produce an initial prediction based on a simple averaging or regressing the data from the reference cell lines. Then we correct that prediction in step number 2, and by correcting, we mean we map the initial prediction onto the gene expression space of the target cell line using the background data. And the mapping method could be either linear, such as the principal component analysis, or non-linear, such as an autoencoder. Here we can see more analytically each step and the respective methods. For the mean method, as was mentioned earlier, we use a simple averaging to compute mean transcriptional signature for a drug at a specific drug lot using the reference dataset. By drug lot, what we mean is we evaluate that at specific drug concentration multiplied by the duration of the treatment. Meanwhile, the regression method, we use all the data of the drug from the reference cell lines to build a linear regression model for each gene separately, where the gene expression value is the dependent variable and the drug lot is the independent variable. That produces an initial prediction. So moving to step number 2, we want to correct the initial prediction. We can do that linear in the PCA projection method. For instance, the pink dot represents the initial prediction here, and what we want to do is to identify the nearest neighbors of the initial prediction with respect to the background data. Once we identify that, these are represented with a green light color, we use them to project the initial prediction from step number 1 onto the gene expression space of the target cell type. And then we have the correct prediction. We can correct the initial prediction non-linear by using an autoencoder. The way we are doing that is we train initially an autoencoder using the data from the target cell type. Then we take the trained autoencoder and we pass the initial prediction from step number 1. The output is the corrected prediction. We can use those two different corrections independently or consecutively. In fact, we found that the best improvement achieved if we use first the PCA projection and consecutively the autoencoder. Now, we wanted to evaluate our method, and to do that, we used the gene expression data from the CMAP dataset for 7 different cell types and 100 drugs, and these have been measured for 978 genes. In addition, our method was benchmarked against an imputation method called tension trade weighted optimization, abbreviated as the TTWPT. So in figure 1, in all subplots except 1C, you can see that we evaluated in different ways the correlation between the actual data and the prediction from the mean corrected consecutively by the PCA projection and the autoencoder prediction. We can see that the methods achieve very high correlation, more than 90%, in the vast majority of the cases. More specifically, in figure 1B, you can see it's 97%. In figure 1C, what we are doing is we compare the correlation between the actual data and the mean method and its all possible corrections, as well as the WPT algorithm. We can see that the best method, again, is the mean method consecutively corrected by the PCA projection and the autoencoder, and on the other hand, the worst one is the WPT. Now, moving on, what we wanted to do is to check if our method can accurately predict upregulated and downregulated genes. For that reason, we used AUPR, area under precision recall, and the URC, area under receiving predicted characteristic, to evaluate our strategy. In figure 2, you can see viral plots that compare our two-step imputation method using original predictions from the mean method and corrected with all possible combinations of corrections, and the WPT algorithm for predicting upregulated genes. These are the 2A and 2B diagrams. And also we did that for the downregulated genes. These are the 2C and 2D diagrams. The accuracy of the prediction is evaluated by UPR. This is the 2A and 2C, and AURC, 2B and 2D. In all the cases, you can see that the best performance is achieved by using the mean corrected consecutively by the PCA projection and autoencoder. This is the light green violin plot, as you can see here. And the worst is the WPT. In table 1, what we are doing is actually we generalize the results that we present in figure 2 across all the cell types of this study. Thus, we calculate the UPR for predicting upregulated genes across all the cell types. You can see that for every cell type, the best AUPR is achieved when we use the mean method consecutively corrected by the PCA projection and the encoder. To summarize, we propose a two-step imputation strategy for predicting the track response gene expression data in a target cell type using reference cell types, and apply the strategy to the CMAP dataset. The main takeaways are as follows. The PCA projection correction combined with the autoencoder correction scheme achieves the best improvement of the baseline method, and all our methods outperform the state-of-the-art method TDWPT. Thank you very much.